Welcome back. Today we're going to be discussing chapter 5 from microbiology on viruses and prions, or basically infectious agents smaller than bacteria, archaea, eukaryotes. In this chapter, we will be covering where they fit into the biological spectrum, the general structure that you would expect to find in viruses, how viruses are able to multiply and infect their hosts, as well as different techniques for cultivating viruses so that we can study them, use microscopy, different methods to count and identify them. And then we'll close up this chapter with other non-cellular infectious agents like prions that were on our last slide, and what role all of this plays in human health. For this first lecture, we're going to be primarily focusing on numbers 1 and 2, how viruses fit into the biological spectrum and their overall structure. All right, so first, where do they fit in the biological spectrum? In earlier chapters, we've discussed what does it mean to be alive? What are some of those characteristics we use to determine if a robot is alive, if we're alive, things like that. And viruses are seen typically as non-living infectious agents because they are unable to reproduce on their own. They require a host. They have been known to infect a wide swath of different hosts, including bacteria. That would be like bacteriophages. They can also infect algae, fungi, protozoa, plants. As you see on the left here, a virus is responsible for producing this variegated pattern in the tulip, as well as animals, as we are all keenly aware from the latest pandemic. For many years, the cause of viral infections was largely unknown. We could put together that, yes, smallpox was transmittable from person to person and polio could be transmitted as well, but we couldn't quite track what was responsible. The first person documented to have made a big leap forward on that front is Louis Pasteur. And we've heard his name before in earlier chapters. To jog your memory a little bit, he was responsible for some early vaccines, as well as if you think back to the experiment where they kind of broke off the crook-necked part of the glass flask to determine if living things brought about living things or non-living things could spontaneously produce life. He was also responsible for hypothesizing that rabies, that we think of with like rabid dogs and things like that, was caused by living things that were smaller than bacteria. And he was the first to develop a vaccine for rabies. Further work on the discovery of viruses was continued into the 1890s when a pair of scientists showed that a disease of the tobacco mosaic virus that caused loss of crop yields in tobacco was actually caused by a virus and was infectious, and it was not a bacteria. The same time, as another pair of scientists, Lofer and Prions, were responsible for discovering that a virus in cattle caused a foot and mouth disease that also was detrimental to crop yields. These early research teams found that when they took infectious fluids, either from the cows or, maybe ground up the tobacco and drained it off and passed that fluid through a filter, the fluid would not trap anything. So we could do this if we had a bacteria that we thought was responsible. And if you passed the blood or whatever fluid through it, it would trap the bacteria and the purified fluid would flow through. But because viruses are smaller than bacteria, they wouldn't get stuck in the filter pores they'd run right through. And so even after filtering it, the fluid was still infectious. This showed that the infections of the tobacco mosaic virus and these animal viruses in cattle were caused by agents smaller than bacteria. Over the next several decades, our knowledge as a scientific community has continued to grow and even grows still. In the early 1900s, the big push was towards understanding the physical, chemical, and biological nature of viruses. So figuring out how they worked, what their basic chemistry was, of what sorts of molecules, biomolecules were involved in them, and what their nature was in the overall biological sphere. We now know that viruses are non-cellular particles. They're non-living, and they're not going to have the same sorts of traits that we've talked about in the last few chapters on components that are always found in a cell. They will, though, be very defined in their size, shape, and chemical composition. So just because they're not technically living does not mean that they aren't highly organized little machines. In the last few decades, the big push has been towards imaging and sequencing of viruses. So we can now image them with electron microscopy and fluorescence microscopy. I've seen some papers with fluorescence microscopy where you can actually track infections of HIV in like a sample, obviously in a dish, not in a patient, but on a microscope, and you can track and see what are the kinetics, how is it moving, how is it infecting its host. So we can image it now, actually watch what's going on. And with the invention of next-gen sequencing, of being able to sequence whole genomes, DNA and RNA, things like that, we've gotten a clearer picture of the different types of viruses, the numbers of them, what genes they have, and we're better able to get a full picture of what we're up against. So here are several properties of viruses that your book highlights. First of all, they are obligate parasites. So that's part of why we characterize them as non-living, is because they have to have a host, be it bacteria, protozoa, plants, animals. They have to hijack machinery, cellular machinery from an actual living cell in order for them to propagate and survive. It's estimated that there are 10 to the 31st virus particles on Earth. So we are going to have 10 with 31 zeros here. Huge number of them, approximately 10 times the number of prokaryotes currently present. Along with that, they're ubiquitous in nature, so meaning that you would find them pretty much anywhere, again, because they can have a wide range of different hosts. They're ultra microscopic in size, so again, we can't see them with a regular light microscope. We have to have further technology of maybe an electron microscope or a confocal microscope to be able to image them properly. They're not cells. They're not going to have organelles or a cell wall or things like that, but they do have structure. They're very compact and efficient and economical with what they do have. You won't find them independently because, again, they need their host to fulfill all the characteristics of life. Their basic structure, which we'll get into later in this lecture, consists of a protein shell known as a capsid surrounding a nucleic acid core. So you will always find that a virus will have either DNA or RNA at their core, and they're going to have proteins surrounding it. The nucleic acids could either be DNA or RNA, but they're not going to be both. And they can be double-stranded or single-stranded. Molecules on the outside of the virus, on its surface, are what gives it specificity and determines which cells they can attach to, and along with that, their host range. So based on what surface markers they have, they can dock with certain cells and not other ones. They multiply by taking control of the host's genetic material and their synthesis material to help churn out more viruses to infect more hosts and so on. They do not have enzymes for most metabolic processes, and they're not going to have machinery to synthesize their own proteins. For those, they have to use the host's machinery. Right, so with that general overview, we're going to focus in on how viruses are classified and how they're named. So viruses used to be classified solely on what their host was and what kind of disease they've caused. And again, as we've learned more with sequencing and imaging, we're kind of rounding out our classification and using more tools, specifically the actual nature of the viral particles. So not just what is their indirect effect that we see in the patient, but the virus itself. The main criteria we currently use are: what is its structure? Is it a naked virus? Is it an enveloped virus? Does it have certain chemical compositions of maybe an extra enzyme? Or does it have certain protein subunits on its outside, as well as its genetic makeup? Is it a DNA virus? Is it an RNA virus? And in the back here, you can see the measles virus. Again, while we can image it, you'll notice that it isn't in nearly as crisp of detail as we would get with